Hi, my name is Subin, and I'm a senior bioinformatics scientist here at Illumina. Today I'll introduce DRAGEN Targeted Microbial, an analysis software for hybrid capture enrichment panels and amplicon assays like IMAP flu and COVID-seq. As input, the app takes FASTQ files consisting of sequencing reads, and as output, it generates consensus sequences out of those reads. Lastly, it also runs Pangolin and Nextclade, which provide clade and lineage information on those consensus sequences. Here's the input form shown when you launch DRAGEN Targeted Microbial. First, to provide the input FASTQs, you either select individual input biosamples or a single input project. If you select an input project, the analysis will be launched for all samples in that project. For IMAP, select amplicon as your experiment type, which allows you to open up this drop-down menu and choose your amplicon primer set of interest. Reference files for these are preloaded onto the app, so you don't have to provide your own reference files. If you want to provide your own reference, you can select Custom and open up this custom reference box. Here you can provide your own custom reference FASTA containing reference sequences for consensus sequence generation. Optionally, you can provide a custom reference bed to define each sequence in the reference FASTA. You can also provide custom PCR primer definition file to describe the primer locations or sequences used in your experiment. Tools like Primal Scheme can be used to generate this file. For all of these custom files, the I button provides details on how they should be formatted. For custom reference, you can also select which Nextclade datasets you want to use for analyzing your consensus sequences at the end of the pipeline. Whether you're using custom reference or any of the predefined amplicon primer set, you can enable or disable Pangolin and Nextclade. Once you're ready, you can launch the application and it'll initiate the analysis. Typically, after a few hours, your analysis will complete and you'll be able to see your output on BaseSpace. Here we'll show an example from running DRAGEN Targeted Microbial on 3 influenza samples. First, here's a summary report with results from all input samples. In the metrics by sample table, we provide a download button for combined FASTA file of all consensus sequences generated in this analysis. Next, we have a table with each row corresponding to a sample. In each row, we report metrics like fraction of detected amplicons, genomes generated, and percentage of callable bases. We also provide links to download consensus sequences, unmapped reads, and de novo sample contigs. In the following table, we show output from Nextclade for all input samples, where you can get useful information such as clade assignment. Report for each sample can be accessed on the left side. Here we show results specific to a single sample. First, we have download links for consensus sequences and contig sequences for the sample. Then we have metrics by virus table, which shows the viral genome generated in this analysis. In this example, we report a single influenza A virus with subtype H1N1 along with genome-wide metrics like median coverage. Next, we have metrics by sequence table, with each row corresponding to a single reference sequence used during short read alignment. In this example, we show all 8 segments belonging to the influenza A H1N1 genome. For each sequence, we provide metrics like percentage of callable bases, median coverage, consensus length, and so on. We also present a breakdown of input reads showing how many reads were removed during QC and dehosting, and also how many were mapped versus unmapped. We also report how many reads mapped to each reference sequence used during short read alignment. Next, we have coverage plots with x-axis corresponding to base position along the reference sequence and y-axis corresponding to coverage depth. Here we show all 8 segments of an H1N1 genome. These coverage plots can be viewed in log scale using this button. Shapes on the plot denote sequence variation, with the y-axis on the right corresponding to allele frequency. You can hover over the shapes to get more information on each sequence variant. Lastly, we have the NextClade table, but containing rows specific to this particular sample. In this example, NextClade was run for the consensus sequences corresponding to segments 4 and 6, which corresponds to HA and NA genes of influenza A genomes. As we demonstrated here, our app is easy to use and provides a wealth of information for analyzing data coming from IMAP experiments. For more information on IMAP, please check out our other videos and reach out to tech support if you have any questions.